Hello learners! In our last video, we talked about DNA, the molecule that carries the instruction for life. But knowing what DNA is raises an even bigger question. How does a cell make sure every single new cell gets a perfect copy of it? That's exactly what we are covering today, DNA replication. It's one of the most precious and elegant processes in all of biology, and once you understand it, you will never look at cell division the same way again. Welcome to Bioinformatics. My name is Komal. Let's get into it. So imagine someone handed you a book filled with 3 billion letters and asked you to copy it perfectly. Zero mistakes, every letter in exact right order. Seems impossible, right? Even the most advanced photocopier would eventually make mistake. Yet every time one of your cell divides, it does exactly this. Not occasionally, every single time. And it does it fast. So today's question, how does DNA copy itself with such incredible accuracy? Let's find out. Why does DNA replication actually matter? Before a cell can divide, it faces a critical rule. The new cell must receive a perfect, complete copy of its DNA blueprint, not a rough draft, a flawless duplicate. Without this, growth, healing, and reproduction completely grind to a halt. Every single new cell your body creates to heal a wound or replace old tissues relies on this process. In short, DNA replication is the ultimate genetic handoff. It is the foundation that makes life and healing possible. So when does all this copying actually go down? It's not random. It happens during a specific window called the S phase of the cell cycle. Think of the cycle like a timeline. The cell prepares in G1, copies its DNA in the S phase, double-checks its work in G2, and finally divides during mitosis. By doing this heavy lifting before the split, every new cell gets a perfect copy of your genetic code. It happens just once per cycle, ensuring your body can grow and heal without a hitch. DNA replication doesn't start just anywhere. It begins at a specific spot called the origin of replication, or ORI. This is where the double helix unwinds, forming a replication fork. An enzyme called the Helicase unzips the strands so DNA polymerase can build new ones, one continuously, the leading strand, and one in fragments, the lagging strand. Here's the difference. Bacteria have just one origin of replication on their single circular chromosome. Human cells have DNA that's linear and much longer, so they use multiple origins at once, creating many replication bubbles. This matters because it lets even huge, complex genomes get copied accurately and fast before the cell divides. Now let's look at the step 1, unwinding of DNA. Before replication can begin, the double helix has to open. An enzyme called Helicase does this job, acting just like zipper pull to break the hydrogen bonds between bases and separate two strands. Once open, single-stranded binding proteins SSBs attach to the strands to keep them from snapping back together. It's important to note that Helicase only breaks the bonds between bases, leaving the strong sugar phosphate backbone completely intact. In short, Helicase unzips the DNA at the replication fork, creating the open template needed for copying to begin. Step 2. Stabilizing the DNA. As Helicase unwinds the double helix, the DNA ahead twists tighter, like over-twisting a rope. Topoisomerase leaves this tension by cutting and rejoining the strands, letting Helicase keep unwinding smoothly. But the separated strands now risk repairing. Single-stranded binding proteins SSBs attached to the exposed strands, holding them apart, like clips keeping two pages open. Together, topoisomerase release tension and SSBs keep the strands stable, allowing replication to proceed smoothly. With the DNA unwound and stabilized, you would think DNA polymerase could jump right in and start copying. But surprisingly, it can't. And here's why. DNA polymerase needs a starting point. It's simply not capable of beginning a new strand from scratch. It can only add on to an existing one. So the cell brings in another enzyme called Primase. Primase synthesizes short RNA primer, which attaches to the template strand and provides a free 3'OH group. And this is exactly what DNA polymerase needs to attach the very first nucleotide. Think of the RNA primer as a starter handle. Without it, DNA polymerase has nothing to grab on and build from. With the RNA primer in place, it's finally time for the main player to step in, DNA polymerase. DNA polymerase reads the template strand and adds complementary nucleotides one by one, building the new DNA strand. But it has one strict rule: it can only synthesize DNA in the 5' to 3' direction, never the other way around. Notice how with the replication fork, DNA polymerase moves smoothly along the leading strand, adding the nucleotides continuously. But on the lagging strand, it works in short bursts. Since it's consistently moving away from the fork instead of towards it, beyond just adding nucleotides, DNA polymerase also proofreads the newly synthesized DNA as it goes, catching and correcting errors along the way. Leading versus lagging strand synthesis. Two strands of the DNA run in opposite directions, but DNA polymerase only synthesizes 5' to 3', posing two different strategies. Leading strands synthesize continuously, moving with the replication fork. Lagging strand moves away from the fork, synthesized in short Okazaki fragments, each needing its own RNA primer. Both strands go to 5' to 3', one continuous, one fragmented, working together for accurate, efficient replication. Remember those short fragments on the lagging strand? They are called Okazaki fragments, and now it's time to complete the picture. First, the RNA primers within these fragments are removed and replaced with actual DNA nucleotides. But this leaves small gaps or nicks between each fragment. That's where DNA ligase comes in. It seals these nicks by forming phosphodiester bonds, joining everything into one continuous strand. Think of DNA ligase like glue, joining puzzle pieces together to complete the picture. So Okazaki fragments allow DNA replication to occur in the 5' to 3' direction, while DNA ligase ensures the final strand is accurate, complete, and efficient. Now let's talk about accuracy and the concept that ties everything together: semi-conservative replication. As DNA polymerase builds the new strand, it doesn't just add nucleotides blindly. It proofreads the newly synthesized strand, catching and correcting many errors along the way. And here's the beautiful part. Look at this model. Each new DNA molecule contains one original parental strand and one newly synthesized strand. This is what we call semi-conservative replication. And it's just not a theory. It's exactly how your cell preserves genetic information with remarkable fidelity, generation after generation. So, semi-conservative replication ensures accurate transmission of genetic information from one generation to the next. So, why does all of this actually matter? DNA replication is not just a textbook process. It's the foundation of life itself. It's essential for growth and development, tissue repair, cell division, and embryonic development. Every time your body heals a wound or cell divides to help you grow, DNA replication is working behind the scenes. But it goes even further. This process underpins many areas of modern genomics and medicine, from DNA sequencing to cancer research, forensic science, and biotechnology. In short, understanding DNA replication is not just about biology. It's about understanding the very foundation of life and modern science. Prokaryotes versus eukaryotes. Prokaryotes usually have just one origin of replication, a circular chromosome, and one replication bubble. Eukaryotes, on the other hand, have multiple origins of replication, linear chromosome, and many replication bubbles happening at once. But don't worry about memorizing all of this just yet. The core mechanism is similar in both. We will dive deeper into the finer differences, including DNA polymerases and telomerases, in the future episode. Let's bring everything together with a quick summary. DNA replication happens in 8 simple steps. It begins at the origin of replication, where helicase unwinds the DNA. Telomerases and SSBs then relieve tension and stabilize the strands. Primase lays down an RNA primer, and DNA polymerase takes over, adding nucleotides in the direction of 5' to 3'. This creates the leading and lagging strand, with DNA ligase sealing the Okazaki fragments together. And finally, the process produces 2 identical DNA molecules through semi-conservative replication. The key takeaway: DNA replication is a highly accurate, semi-conservative process that faithfully copies genetic information before every cell division. So, how does all of this connect to what we do here at Genomics Informatics? Every genome analyzed by a bioinformatician begins with one fundamental process: DNA replication. Understanding this process gives you the molecular foundation for advanced topics like whole genome sequencing, next generation sequencing, variant analysis, transcriptomics, and metagenomics. Here at Genomics Informatics, we'll continue connecting these biological concepts to real-world genomics and bioinformatics applications, because mastering DNA replication is truly the first step to mastering genomics and bioinformatics. That's it for today. But here's something to think about. How does 2 meters of DNA fit inside a nucleus so small you can't even see it? Find out in episode 3, Chromosomes Explained. We will watch DNA wrap around histones, form nucleosomes, coil up tighter and tighter, and finally pack itself into a chromosome, shrinking down to just 6 micrometers. Hit subscribe and catch episode 3 only on Genomics Informatics. Follow us on Instagram for bioinformatics tips, learning resources, and updates about upcoming videos. Stay curious, keep learning, and keep growing. Thank you for watching, and I'll see you in the next video.